Welcome to another episode of the Dendrite Podcast with Saeed and Farnam. This is a conversation with Dr. Jonathan Lee. He's a professor at the Department of Biochemistry, Microbiology, and Immunology in Faculty of Medicine at uOttawa, and the director of Translational Molecular Medicine Undergraduate Program. During his scientific career as a researcher at the University of Toronto, McMaster University, and University of Ottawa, he has focused on the biochemistry of immune cells, breast cancer, and the molecular biology of cell motility.
Besides directing the TMM program and teaching, his current research projects are exploring the involvement of mitochondria in cell adhesion. He's also taking a new approach to noises in high-throughput genetic data to identify biologically important genes based on their level of variance. We hope you enjoy this episode, and now here's Jonathan Lee.
Hello, Dr. Lee. Good morning. Thank you for joining us here. It's a great honor after the class and having you now just have a conversation, because we had a lot of, like you mentioned, a lot of conversations. Just we would ask questions and you would have, look, this is a conversation you have like with that researcher or that prof. And now we're sitting down and having this conversation with you.
Well, it's a pleasure to be here. I look forward to this. I think it's going to be fun.
So maybe we can start by you telling us about your journey when you were an undergrad at Western, right?
Yeah, so it's been a long journey. So I started my undergrad in 1983, before there was such a thing as internet. So it tells you how far back that was. And then in 1987, I went to Toronto to do a PhD in immunology. Right. Then I did 3 years of postdoc with Alan Bernstein, also in Toronto. Then in 1995, I moved to McMaster University. And in 2003, I moved here to the University of Ottawa, where I've been for the last 20
You've been
plus
all
years.
across Ontario.
Yeah, so
Which city is your favorite? I'm sure like Ottawa is the coldest, and I don't like Ottawa at all.
Oh yeah. So each city has its different pluses and minuses. Yes, Ottawa is colder than Toronto is, but you know that my family is a skiing family, and so that Ottawa is a great place to do that. It's a nice, relatively small, relatively friendly city. I do, on the other hand, miss Toronto and its vibe and its restaurants and its so many different cultures within an accessible distance.
Right, yeah.
So, I mean, of the cities in Ontario, definitely Toronto and Ottawa.
Okay. At least we got it.
You got that one for Toronto, right?
Okay, that's good. So how did you become interested in cell biology?
Well, so that's kind of a long story, and so that my training as a PhD was as an immunologist, and it really was the biochemistry of immune molecules. And then I became a cancer biologist as a postdoc, and when I moved to McMaster, I was really a cancer biologist. We used a lot of mice, and we were studying tumors, but you know that part of the things about science is you kind of follow your nose and you go where things lead you, and amongst other things that we found a regulator of the actin cytoskeleton when I was in McMaster, and so that we followed that through. And since that time, my lab has primarily been about cell biology and about the biology of movement.
So fascinating, because of having like flashbacks of the classroom covering all this stuff.
Well, I'm glad you remember it.
Yeah, that was the goal. So
Was he a good student?
I think he was. I don't have his mark handy, but I think yes.
Well, the fact that you remember my face, I mean, from
Of course.
that on its own should like.
TMM is a small enough cohort that I recognize faces. This summer, there was a TMM student who graduated in medical school and was starting family medicine, and he was just out there. There was a barbecue, and so I recognized him. His name is Mohammed, and I thought, oh, well, what are you doing here? It's been 4 years since I saw you last. So TMM is small enough that I recognize most of the faces in the English stream. The French stream less so, because I don't actually teach in the French stream.
Right. Yeah, we have a lot of questions about the TMM itself.
Okay.
Let me just ask you, so you know how some people say you gotta set goals for yourself and know what you're doing in like 10 years. But again, like you don't really know what's gonna happen to you. You just gotta follow your nose and see where it leads you. What do you say about that? Like, do we need to set a goal for like at least 5 or 10 years? Be saying, oh, I want to definitely do this. I'm gonna achieve that, and then. But again, like it comes down to like you don't really know what's gonna happen. So which Which way do you choose?
Yeah, so personally, I've never had a 10-year goal because as you rightfully said, it's just too far down into the future to really make plans. So, but for me, 5-year goals were always important. So that when I got into the PhD program, I knew I wanted to graduate in 5 years, which I did. When I started my postdoc, I wanted to be a university professor within 5 years, which I did. And then after you become a university professor, then there are these timelines of doing something. So that in 6 years you get promoted, 6 years after that you get promoted again. And so that there are these goals to have, but if someone had asked me when I was a graduate student what kind of science I was going to do in 10 years, I would say, well, I don't know. You just keep following that through.
Right.
So I think it's important to have goals, that when students come into my lab, I tell them to have short-term goals. And so that's something I want to do within the next couple of weeks. Midterm goals, meaning something I want to get done in the next 6 months. Long-term goals, when am I going to graduate? What am I going to do afterwards? So that it's important to think about those things without too much emphasis on, in 10 years, I want to be this, because things change.
Yeah.
So what is your experiences with, because your career, I think, has been really well sphered in teaching and research and coordination and all that. So what is your experience with setbacks or difficulties or challenges, or just getting the hang of the new things that you have to do?
Yeah, so I mean, as scientists, that most of the things we do in the lab fail. 90% of the experiments fail. 90% of our ideas are wrong. So you kind of get used to that. And you also get used to rejection, that you write a grant, it gets rejected. You write a paper that you think is great, it gets rejected. So that setbacks are just part of being a scientist, and you get a thick skin because it's going to happen. That there is no one that I know of who's had every grant accepted, who's had every paper accepted, and whose ideas are great. Setbacks are part of it, and it only makes successes better.
Right. Can you explain what's the process of submitting a grant, and then who decide to give you the grant or not, and why they reject it?
So I sit on a bunch of grant review panels, and so obviously I've also submitted probably hundreds of grants in my scientific career. And so that there are different agencies. There's some federal government agencies for grants. CIHR and NSERC are two of them. Then there's some charitable foundations that give out grants to researchers. The Canadian Cancer Society, the Cancer Research Society, other health-affiliated grants. And so the way that this works is that these agencies have a grant form that you need to fill out with a description of the proposed research, send in your CV. Then it goes to a panel where typically 2 or 3 researchers read your grant and then decide whether it's exciting and worth funding or not. And unfortunately, the way this works now in most of the panels, that somewhere between 80 or somewhere around 80% of the grants that are submitted get rejected. So that's, and that panelists spend a lot of time reviewing grants, and most of the time we try to be supportive. What would it take to get this funded?
Right. Now I'm sure it's hard for them as well to choose and be able to
Yeah, I mean, with funding success rates that are around 10 to 20%, that means that the majority of people are going to get rejected. And so you're separating the superb from merely excellent. Right. And that's very hard to do.
Hence all the proposal assignments and figure assignments that we have to
yes.
we have to do part of our courses. So what are your current interests?
So my current interests, so I have 2 really. So one is to continue to look at cell adhesion, and we have some data suggesting that mitochondria are involved in cell adhesion, which is quite exciting. And we're also involved in some computational work, which is something that I started with Ted Perkins about 10 years ago. And the idea is to think about noise in high throughput data, not as something not as something bad, but as something indicative about the gene itself. So that variance across a population for a given gene, we think, is an indicator of something interesting about the gene itself.
Like it's not just like the background noise that we have to like disregard. It's actually something that we have to look into.
Yeah. So, typically when people do an experiment, let's say that they're looking at the expression of gene X under two different conditions. And so the way that you would determine whether that difference between those two conditions is that you make a p-value assessment, which depends on the variance of the gene expression under X condition and the variance in gene Y. And if the variation is really high, then you can't make a statistical conclusion. But if you look at all genes across all people, for example, that there are some genes that have naturally high variance across a population, and there are some genes that have a naturally low variance across a population. And so that itself is interesting because not all genes, so this is not an experimental error. It's about how gene regulation is controlled. And at least one of the ideas that other people, and myself and Ted included, is that the lower the variance of a gene, the more important it is. The higher the variance, the less important it is. And these things also change around depending on cancer states or disease states. So we think that there's something interesting here with variance within a population.
Right.
I guess the interesting part would be like how to like approach it. So that's one good criteria to like the less variant is. The importance goes up, but then like what to make of it, that all like comes.
Yeah, so we've tried mapping variability and variance based on gene annotation, whether it's an essential gene, whether it's a gene that's associated with aging. And so we don't have full conclusions about that, but it's certainly a work in progress.
How is the balance between the different roles that you have, like at uOttawa?
So I have three roles. So that I teach, I run a research program, and I'm also an administrator of TMM, so that I'm the director of TMM. And so that those three jobs are very, very different.
And demanding.
So research obviously is my bread and butter. It requires management of people and getting money and having creative ideas and troubleshooting. Teaching is actually, it's not easy, but it is relatively straightforward. I come and I lecture, I make sure the content is up to date, make sure that people understand that. It's also really fun because it's not like I'm going to get rejected teaching. And then administration. So administration is a part of the world that you students don't actually see. And so that's a lot of work. It can be frustrating at times. Because I don't get all the budget I want, I don't get all the space I want, I can't get all the resources I need. So that can be frustrating, but that I think that the TMM program is a good program, that this is a program that's been built over the last 10 years to be one of the key programs at the University of Ottawa that's enjoyed by the students. So that makes me very happy, even though it can be frustrating at times as an administrator.
So I guess the lab work would be your favorite amongst the three, right?
Yeah, because you get to interact and solving issues and discovering new ideas and having them, or thinking new ideas and having them confirmed experimentally is very, very satisfying.
Right. Speaking of TMM program, how did it got started in the beginning? Was it like an idea or like a research lab that decided to the students?
I think it started in just over 10 years ago. I think the first cohort was 2016, or is it 2015? Maybe it was 2015. So over 10 years ago, that there was a general dissatisfaction amongst the faculty of medicine people here with the undergraduate education downtown. Big classes, and really a curriculum that didn't reflect the modern biomedical research that went on here in the faculty of medicine. And so the idea of TMM was to train students and to give them a taste of what research science is really all about. And so that's why we want people in the lab doing fun things all the time. And the program really was created under the idea that what would you like your graduate students to know before they got into graduate school? And so that's what is the central theme that runs through TMM. So things have changed so that, science communication 10 years ago was about writing papers. And now it's about writing papers still, but it's also about making podcasts, making videos, doing an elevator pitch. All of these things are part of communication because that's part of what scientists do all the time. So things change and modernize, and that's something we're able to do because the Faculty of Medicine controls this course.
I'm just wondering where it leads with the whole AI system and technology. It's definitely going to be really cool.
Yeah, so AI is going to be a challenge. Obviously, there's lots of good things about it, but in terms of assignments, that you could go to ChatGPT and say, design an experiment like this. Most of that, you can tell that ChatGPT wrote it because it's hyper detailed in one things and poorly detailed in others, and it doesn't go together. So that AI is going to be a challenge as well as a benefit for programs in the future.
Yeah, the whole with AI, I feel like the only advantage humans have is sometimes being imperfect, because and there's a value to that as well. You can't be perfect all the time, and you'll be missing out on a lot of things. So we'll look forward to the...
Yeah, so AI does need to improve because AI can be frustrating at times. That if you've ever been on a help site and you've got a chat bot, there's sometimes that because sometimes it's very helpful. If there's something simple, then the chatbot will be good at it. But if you have a complex problem, the chatbot doesn't know what to do, and you get stuck in this circle, this loop, and it's I want to speak to a human being. So AIs will obviously continue to develop, and it'll be a benefit as well as a danger for a lot of things.
Yeah, there was this tweet this morning. Between Elon and the founder of OpenAI.
Yeah.
So they said they have a lot of money to fund the whole program, but apparently Elon said that they don't have the money. There's a beef going on. We'll see where it leads.
Yeah.
Well, back to TMM now. So what was the challenges of creating a new program? I guess there's a lot of administration.
There's a huge administrative burden to create a new program.
Have you been since the beginning, or?
Yeah. So the thing about TMM when it started that we had a much smaller number of courses than we have now. The first cohort in TMM was only 16 students. Now each cohort is 80. And so every year that I've updated the courses, and that's part of the administrative burden. So we're constantly updating the courses, updating the requirements, and then we've created these integrated programs where TMM is integrated with a master's program or integrated with a PhD program, and so that we've created these things, and we'll continue to do that as things go. It was a lot of work to create TMM. Daniel Figues and Jean-François Couture were the founders of it. And it was a lot of work to get this together. And everyone had to create new course material and create new courses.
Right, and I think every year it's growing to more students apply, and you guys have to
yes,
to deal with that.
Absolutely. Right now we're capped at a total enrollment of 80 just because that's the capacity of the lab that we have. But last year we had almost 200 applicants.
Right. 80 for English or French?
So in total. So typically it's about a 20% francophone application. So probably, I don't know the numbers, but probably 160 English and 40 French. And we admit last year we admitted 21 French students and we actually admitted 60 English students.
So you said it's a great program and it's definitely going to help people for their graduate study. What are some qualities that you're looking for students when you're trying to evaluate them to get into this program?
The first is that we want them to be good communicators. We interview people who meet the course requirements, and so basically the idea is that that TMM is not just about memorizing the Krebs cycle or knowing the PK of histidine. It's about the ability to present scientific data, to understand it. So communication is one important skill. Curiosity is another one, because science is really about discovery. And so the third year lab is you isolate and name and characterize your own bacteriophage. So that's a very unique lab that's run by Adam Rudner, and everyone loves it because it's about discovery. If you want to do discovery, you have to be curious. So curiosity, as well as communication, are important, as well as that the course load is heavy. So it's not an easy thing to do because there's a lot of stuff that you have to learn and unlearn. And there's a lot of material to cover. Third year, the first term there's a lot to cover between molecular biology, biochemistry, and my cell biology course. There's a lot of new information that gets dropped on you very, very quickly.
Yeah, because apparently there's a myth about TMM is easier than biomed or other studies. But
Well, I don't think it's easier.
Maybe it's more fun, I guess.
It's different in a lot of ways. Like the focus, I mean, I don't know the third and fourth year of like the other programs, but in TMM it's much more that, okay, now you have to think, you have to make a proposal, you have to there's a seminar that you have to think about, there's papers that you have to read. There's a lot of other things, as you said, like on the research aspect that we have to cover. And on the slides as well, it's much less not that it's not based on text, but it's like papers that coming from like all the other faculty members and all the other people in the field. So
So different is the best way of doing it. Now our marks do tend to be high, but then that's because we recruit students with high marks. That's no guarantee that their marks will remain high.
Right.
But it's a different type of learning. So in my cell biology course, that we put you in a team and you make a 5-minute YouTube video.
the first assignment.
That's the first assignment you'll get in the English stream of TMM. And so that's an unconventional assignment, and it's hard, but it's a different hard than memorizing the pKa's of all the amino acids. It's someone's got to write a script. Someone has to run the video, someone has to create graphics, someone has to research the content. All of these things are hard, and they're different from the other things that are in other programs.
And what goes in, I don't want to say like recruit, I don't know what the correct word is for it, but choosing the different professors, lecturers for every different course, is it like just similar to any other program?
Well, we try not to assign courses to people. So the courses are created based on the expertise of the person who's in it. So that my cell biology is very heavy on cytoskeleton, on signaling, whereas because that's my expertise as a scientist, and also Katie Rayner, who deals with cell death and with lipid metabolism. That's her part of the course. The French version of this course, 3504, which is taught by a yeast guy, so there's an emphasis on yeast in Damien Damour. So that every professor brings their specific expertise to the class, because that's what we want in TMM. So the neuroscience course is taught by neuroscientists. The cell biology course is taught by cell biologists. The biochemistry course is taught by biochemists.
Could you explain a little bit of the whole elements of this program? So I guess first semester is going to be like lecture basically mostly, and
for the most part, and then your lab. So the idea is that we want to give you a basic theoretical grounding in the things that you'll need in the future. So that if you're dealing with mammalian cells, you need to learn how signaling works, and so we spend a couple of lectures on the MAP kinase pathway. Which I hope you still remember. Okay. So that's important, because these are things that in the future, when you read a paper and they talk about, say, GRAB2, you'll know that, hey, I know what GRAB2 is, all right? And I know what GRAB2 does, because it won't be explained in the paper, but these are things that you'll keep in mind. Or, when people talk about the caspase cascade and you read a paper about an anti-cancer agent that targets caspase 9, for example, then you go, oh, I know how that would work. And so these are important things that people will learn in the first term. And in the second term and in the fourth year, then they'll be more specialized in the field that they're interested in, and hopefully take that background knowledge with them to the labs that they work in for their honors project or during the summer.
Okay.
So why did you decide to take part in this program? Like, what does it mean to you? And
So
the level you're involved in it.
So the idea of me becoming the director. So initially in the first 3 years of the TMM program. I was just a faculty member participating in it. And I became the director as part, the idea was to grow the program so that over the 6 years that I've been the director, we've tripled the enrollment and to enhance the capability. So we now have a dedicated microscopy lab. We have lots more fourth year methods courses than we had before. So my goal in becoming the director was to make a bigger program and a better program. And so for the most part, I think that we've been successful at that.
Oh yeah, Said always tells a lot of good things about this whole program.
And also about your own course, like he knows a lot of, like, because he, we were communicating as the semester would progress. And also I wanted to ask you a question about your, like, teaching style as well. Like, is that just comes naturally because...
Well, you know, so one of the odd things about being a scientist, a teacher, and an administrator is that I have absolutely no formal training in teaching or in administration. So this is the kind of thing that you learn as you go along. Like someone just dumps you in a classroom and then you teach. And like I didn't consciously establish a style. You know, basically what I try to keep in mind is that, like probably everyone listening, we've had good lecturers and bad lecturers. And my only fear is being a bad lecturer, so I try to prevent the bad lecturer thing. And so everything else is just teaching the stuff that I know and that I like and responding to the class. When you look out at the class, if you're not making any sense, you can see it in their faces. And then you go, okay, we'll stop. Let's do it again. And I also feel it's important to have participation. If you don't understand something in class, put your hand up, ask, we'll go through it. And that's also important because it's important that you understand the material, not just because the exam's coming up, but I want you to understand the material.
Being evaluated on the material is never fun, but in the class itself, it's like I'm listening to a story. It's like you're going all over the slides and, you know, everyone asks questions. And in that particular lecture, with cell motility, with Rogov, the student, the scooter guy, you know, you got him down as like to play the role for monomers, and then it's pushing the cell. So that will stay. No matter
So these things, part of my teaching style is try to involve people, like the actin monomers creating a polymer and getting it to move, and so that doing something different rather than just showing, okay, here's an actin monomer and here's the KD and this is how it, like, just to think about it in a different way. And so like I added, network biology to my cell biology lectures this year. So network biology is a mathematical thing, and if you put the mathematical formulas, everyone's eyes will glaze over. But if you think about network biology in terms of the things we know, YouTube, TikTok, Facebook,
it's everywhere.
That's everywhere.
Makes sense. Yeah.
But organization principles in the cell have analogs in the way YouTube is organized or TikTok is organized, and what does it mean to be a TikTok influencer? And what's the analogous cell biology thing to a TikTok influencer? So those are things that people understand.
Right.
Whereas if I were to say that, okay, here's a molecule that is a network hub with high connectivity, people would go, oh, well, what does that mean? But I say this molecule is a TikTok influencer. It has, won't have 1 million followers, but it might have 20 followers, meaning 20 proteins that bind to it. So it's an influencer in the network.
Has anybody write TikTok on the exam? Like it's like a TikTok.
Well, so this is part of this. This is part of the lecture because it's a way of thinking about abstract mathematical concepts in things that we understand. So that the abstract principle of connectivity, which is the number of edges that you have with a particular node, is really the number of followers that you have in YouTube or TikTok. And I think one year we had someone with over 1,000 TikTok followers.
Oh, wow.
Yeah, so I have none because I don't do that.
So what is the feedback or the experience of the professors and researchers involved in the program so far?
So far the professors are pretty happy that it's a different type of teaching, and the classes are small overall, so that makes it easier. Like when I taught downtown, even in fourth year classes, I had 300 people in my class in fourth year. And that's miserable because you don't see anybody. I don't recognize any of their faces because there's 300 people sitting in the auditorium and I don't know who they are. And it's hard to, and also people are intimidated. They don't like to ask questions. And so that overall, the faculty are pretty happy. They don't have to go downtown to teach. They can stay up here. The worst thing is a 10-minute walk to Peter Moran. So overall, the faculty are happy that they change their courses all the time to reflect the progression of their own research program. And so it's nice to be able to teach from your research rather than cracking open a textbook and say, oh, today we need to cover, or next week we need to cover chapter 32.
Right. No, this is great. Like the one thing that I really like about TMM, and Said always tell me about it, is the community and the people you have. Like sometimes it might be harder to like go, as you said, like do those assignments, make a podcast or all of that stuff. But at least like you meet with people that you're just gonna have like a long relationship or friendship.
Yeah. So that there's lots of group assignments, and I think that there's at least there's a bunch of couples from TMM that have been created. And I think from the first cohort of TMM, there was a marriage 3 years ago, I think. There may be more, but I know of at least one. So that's part of the community. I mean, there's 80 people in total that are enrolled, and you guys are all together. You're all doing the same labs, the same required courses. You're all in the communication course together. So that's one thing that differentiates us from downtown, that there's a community. You'll know your professors. I'll see you all the time.
The professors also do know each other, because I remember like on many occasions you would ask a question and you're like, well, this is not on my side. You can go have like a year, but like name the professor and have
so
like a 2-hour discussion on this.
Absolutely, right? So that if you want to talk about chromatid, the anaphase in yeast or mammals, like talk to Adam Rudner. If you want to talk about formins and the cytoskeleton, talk to John Copeland. Because I know that the research interests and strengths of nearly everyone in the TMM program, and that's true with everyone else too.
Do we have like a most popular prof at TMM that you?
I would bet that that's Adam Rudner or Lisa D'Ambrosio or Keith Wheaton. So that these three people do the most teaching in TMM, and they invest a great deal of time into TMM, and so that the three of them probably are rotate through my favorite prof.
Okay. Which one is your favorite?
Well, I still haven't covered the whole thing. The list. The list is growing.
So where do you see the whole program in like 5 years, as you mentioned? You said
so that we still want to expand to make. Right now we're limited by space, and so you can see next door that they're building a new building, and so the new space in there will increase the amount of teaching space that's available here. So at one level, we wanted to make TMM a 3-year program rather than a 2-year program. So we would take students after their first year in downtown or other university. And then we still want to keep the enrollment down to a manageable level. I would say that keeping the entry cohort at 100 or less would be a good goal for us, but make it a 3-year program. So that gives us more opportunities to create more labs, to have more advanced content, and to do more like literature reading and hands-on experimentation.
Right, okay.
The number is tricky because, well, you want more people to have this experience, but at the same time, as the numbers increase, well, the community is compromised, sort of. It's like, I guess, like an eternal...
Yeah, so right now, 80 people is good. I think 100 will be good as well. I mean, these are the things we can try, and if 100 is too many, then we could think about reducing the entry cohort. But 80 is fine. The first stage in this will be to make it a 3-year program, and then we can worry about how many students can we properly handle while keeping the TMM experience.
Right. One thing that I always wondered was, so in biomed or like downtown campus, you get to choose to do honors or labs as well. What is the difference between doing that or doing TMM instead and be involved in the lab in TMM?
There's more hands-on training in TMM than in other programs, so that there's the discovery lab in third year, and also we have a bunch of computational labs in the winter term of third year. So that's Adam Rudner's genome annotation course, and also that we have Martin Pelchat and Mathieu Lavelay-Adams' bioinformatics course, which is really about how to get some fluency in R. And so the computational component, and also in fourth year, we have these advanced methods lab courses. So if you want to use a flow cytometer or learn how to do protein crystallization or use a microscope or learn how to do some microbiology stuff, we have a lab for that. Most programs stop offering lab after third year, but we continue to do this so you can get advanced training in these important aspects of science.
And the other part of it is that I think it comes easier. Like on the other side, well, you might be able to get all of this, but you have to do a lot as well, do a lot of searching. But here, since it is the community, like in one single class, well, I'm introduced to 3 different researchers, and they also do sort of know me, and it's like the communication, and to have this opportunity, it comes much easier and it's more available. So
yeah, so you know the fact that you're not an anonymous person in TMM and the faculty aren't anonymous, that's an important part of this. So that if you want to talk to me, you can just walk in. You don't need to make an appointment. You could just like knock on the door and say, "Hey, I don't understand this." It's like, come in, we can talk.
Right. What if a student wants, because usually if you want to do honors, you have to find a professor or find your lab a year before. What if you apply for that position and you want to do honors in your 4th year, but you get into TMM as well? Can you continue working with that prof in the TMM program, or does TMM has its own honors?
The way TMM has its own honors, so that if you're in the TMM program, that you need to take a TMM honors, but we allow students to take their honors project in a lab that's not in the Faculty of Medicine.
Okay.
So that we've had people who've done an honors project in the engineering department, who've done an honors project in the physics department, and in the biology department as well. So that it's not required that way. Now, the other way, someone from downtown can't take the TMM honors project, but they're free to work in the lab of a Faculty of Medicine faculty member. So that the honors we're allowed to take students in the BPS honors project or the biomed or the biology one, so that you can come here and work here without being a TMM student.
Right. Can you do your honors in your 3rd year as well once you're in TMM?
If you're in TMM, no, that's not possible because you need to go through all the 3rd year stuff first.
how is the name decided? Is it because like everyone asks me like, what is your program? Go, like it's TMM. Like
so when it was created, we had a number of different names, and this was the one that the leadership in the Faculty of Medicine decided on. The idea was to capture that, you know, we're dealing with molecular stuff, but also it's somehow related to human health in medicine, and then translational. So translational work means that you're bringing basic science and fundamental research into a clinical setting. And so the idea of TMM was to capture all of that.
Then it reflects that as well. Like every single time I'm saying, well, translational and molecular medicine.
Yeah.
Wow.
So I mean, you know, you look at the treatments; those all represent translational work.
Mm-hmm. And now more on the classroom. What is like a non-academic thing that you'd like people to walk away from your class? Like if it's a heavily scientific class, but from my own personal experience, you do like put in some other stuff in the middle depending on the occasion.
Yeah, so I think the idea is that when is to think about a process as the components of that process, but there are organizing principles in a process that apply to other processes as well. So like a good example of that is the MAP kinase pathway, which begins with a cell surface receptor, then a hormone binding to a cell surface receptor, the assembly of molecular complexes around that receptor, and then phosphorylation events that eventually move a kinase into the nucleus, which phosphorylates a transcription factor and activates transcription. And so that's a process, and all of the components of that process are part of my class. But if we think about another process, so let's say cell adhesion, many cell adhesion processes work in the same way with a transmembrane receptor, molecular complexes that assemble, kinases that assemble around there, they phosphorylate other substrates. So that understanding the components as well as the system allows you to translate your knowledge, because the systems can be applied to different things. The components may be different, but the same organizational principle is there. So that whenever we look at a process, there's a higher level of structure over and above the components, and that's something that I hope that people can appreciate from the lecture.
Make the translation again from like different. The system is the same. It's
the organization is the same, but the components are different, right? So that, like, if you look at let's say a Honda automobile versus a BMW automobile, they share no components, but they are assembled in a very similar way.
Right. Yeah. Interesting.
So, as you mentioned a lot in your classes, you're a big movie enthusiast. Many of the lectures would start with like one or two references to different movies. So, I wanted to ask, like, what is your favorite movie? But that's a harsh question. So
that's a really hard question.
You can
like
choose 5.
So 5 favorite movies. So different genres. So I mean, I think so. My movies tend to be more like action, science fiction, anime. So let's think about an action movie. There's an old movie called Ronin, which I actually love. It has Robert De Niro in it.
Yeah, I've seen it.
Okay, and so that's a really good movie that has some really great car chases, and there's some great dialogue and a great atmosphere. So that's a really good one. So war movies, probably The Thin Red Line, which is a movie. So these are older movies. These are movies from the 90s. It's a Terrence Malick one. Sci-fi movie, it'd be a toss-up between the first Blade Runner and with a more recent one called Arrival, which is a really fabulous movie. Anime, older one, probably Spirited Away. So I'm a Miyazaki film. And then drama, so that's a really hard one too. So drama recently, I don't know, maybe The Florida Project would be one of them. So that the reason I like movies so much is that it allows us to learn things and experience things without being explicitly told them. So a movie is a 2-hour lecture of sorts, but it's not really a lecture. Like, you can learn things from movies the same way you can learn things from reading books. Right. But movies are a way to do this without feeling that you're being lectured to.
Mm-hmm. Yeah, like the whole thing about watching a movie, and because I've been reading some stuff about placebo effects and the fact that we know movies are not real, but still we get emotional and they have their effect. So there's like a relation between that and the placebo effect, and how everything's kind of work out together to, even though they're not real, still they have their effect.
Yeah, absolutely. So I mean, one good example of that is an animated film called Ottawa. I don't know whether you've seen this.
I have it on my list.
Okay, so Ottawa is about a company that's breeding delicious pig-like animals that happen to be sentient, but are nonetheless being sent to the slaughter. And so it's actually a sad film, but I suspect that people who watch that film will actually think about becoming vegetarian just because of the content of the movie. And it's not like the movie explicitly says don't eat meat, but it's an emotional pathway to people think, oh, you know what, maybe cows are sentient in their own way, so maybe I won't eat cows anymore. So
with the animated movie Chicken Run.
Chicken Run. So that's a really fun movie, right? And so that's, you think about chickens and they want to be free, and so that's also a good one. But, you know, like I love movies, and that's why there's lots of movies in my class, scientific movies. And so that over the past 20 years that we've developed the ability to look at cells and processes in sequence. And so that you can learn so much about cell migration by looking at a cell moving, forming adhesions and polymerizing against actin in speeded up time. So you can see it actually happen. And that's it's a better teachable thing than putting it up on a PowerPoint slide and saying, here's actin, here's adhesions.
You can actually see it. Like,
You can see it happening. And it's, you know, the first time I saw a cell biology movie, I was like absolutely like blown away. And then that was probably about 15, 20 years ago. And we've become so much better at live imaging of things.
Mm-hmm. It's also like another sort of, like you get much more motivated because like you're looking at it in a textbook and even images. But when you see the cell moving, it's like it has a personality now. Like you see the phagocyte just engulfing, like you want to root for it.
Oh, you're rooting for it, right? You're rooting for the phagocyte or you're rooting for the cola.
Yeah. So let's say like after a long day, or either a lab or teaching, what's your way go to like relax? Is it like watching a movie or is there anything else?
So one thing I do is that I live close enough here that I can walk to work. So I enjoy the walk. Okay. And so it's a 20-minute walk each way. And so that's a good way of kind of winding down. I'm alone. So I can just like shed the stresses of the day and I can get home. And the other thing I do is I cook. So I feel that cooking is like doing a scientific experiment.
Well, it is. In many ways.
Because you've got a recipe, and then you got to prepare the ingredients. You've got to do something to make something, and you have to improvise along the way. And then you have to do multiple things all at once because you're trying to get it get the rice going, get the vegetables cut. So cooking is another one of these stress relievers because you're doing something with your head and your hands, and the experiment only lasts for an hour or more, and if it fails, well, then you just like.
What is your favorite dish to cook or to make?
It's a variety. So that in my household, that we tend not to eat sequential dinners of the same ethnicity food. So that we'll go through like Chinese, Indian, Western food. We'll try Korean, and so we'll cycle through these things.
Interesting. And my favorite is definitely Italian and Persian because I'm Iranian, of course.
Okay.
Love my mom's food, but also Italian.
Yeah, that's what it does.
Yeah, so you know and sometimes that we're just too tired to cook, so you just like make an omelet or something.
Mm-hmm. Even an omelet can be like perfect. It's not an easy thing.
Exactly.
To take it like make it.
Yeah.
So one of the questions was like how your personal life and your professional life connected. Like I wanted to say like. Who is Jonathan Lee outside his roles in university? But you have lots of roles in university that I think we can like see who you are.
So I mean, so in terms of who I am, so that I have, I'm married. I've been married for 30-something years now, and then I have 2 kids. So that I'm a father. So the kids are older now. That one of them is a surgical resident, another one is an officer in the Royal Canadian Navy. So both of them have moved on from us. I'm a dog owner, okay, so that tends to bookend my days, to walk in the morning and to walk in the afternoon. I bicycle, so I'm an avid bicyclist. I do renovations, and I like to do a lot of stuff with my hands, cooking and renovating. So that's the personal thing. I also garden with my wife. We have a plot over in Kilbourne, so where we grow our own tomatoes and cucumbers, and that's all very, very satisfying.
Right. What type of dog do you have?
Well, we have the dog that we have right now. That we had an older one who passed away. So right now we have a dog named Louie, who's a sheepadoodle. Okay, so that is a sheepdog poodle cross. Yeah, it's really fun.
How old is the dog?
It's 4 years now.
Okay, so sort of still like...
Not quite a puppy.
Yeah.
Thinks he's still a puppy, but doesn't have puppy energy anymore.
That's fascinating. And So now more like a philosophical
Yeah.
question. So what are the things that drive you forward? What is the meaning of life in?
The meaning of
Yeah.
life. Now that's a really tough one,
that
right?
is a
And so I think the meaning of life, things like really change, I guess, depending on what stage you're at, right? So that, 10, 15 years ago, what would be the meaning of life? It's just like, let me make sure that my kids are out of the door and we can help them being successful. And then now sort of the meaning of life is, let's try to remain healthy, exercise, do cultural stimulations, read, watch movies, travel a bit. So that it's changed, right? So that if you'd asked me this 20 years ago, I would have said, I'm just going to get much grant money as I can get and hire as many people as possible and have a big lab. All of these things do change. And so now that I'm close to retirement, that the priorities have changed, that I want to, right now, is to make sure the TMM is in good hands and is well organized for the future. And then now is to really to look forward to other adventures other than the university.
Yeah, there's a quote by a scientist, I forgot his name, but he was saying basically you gotta have a meaning for death as well once you get closer, and then it kind of conclude your meaning of life. It's kind of sad, but it's sometimes like...
No, I think that's fair. My parents both passed away in the last 3 years, and my parents-in-law also did in the past 3 years. And so that part of getting older is cognizant that lifespan is finite. So when you're 30, before you're 30, you always feel like you're immortal, which is a good thing to feel because you take risks and you do things that, like, bungee jumping that most older people wouldn't do. And so that having your life bookended by birth and death is an important thing because it means that the time in between is something that needs to be savored and enjoyed as well as worked on. And so that as you do get older, you become cognizant of the fact that you hear that ticking clock. And what is it that I really want to do in the time that remains?
Do you ever want to do bungee jumping, or have you ever tried?
I've never tried bungee jumping. I'm actually a little afraid of heights, so that even like when I go to an exhibition on the Ferris wheel, I get kind of like antsy on the Ferris wheel. So I'm not a bungee jumping guy. That I promised my wife that we would go parachute jumping at some point, and that.
He did jump out of the plane.
Yeah, I did skydiving and bungee jumping last summer, and it was the best experience.
Okay, so at some point, maybe this. So I made that promise to my wife just before COVID started, and so that of course everyone shut down, but it's time now to revisit the skydiving challenge. Definitely, and also apparently the bungee jumping place that's near Wakefield. Is really good. The one that's in the quarry with the crane.
Yeah,
I've heard it's good.
Yeah, even the skydiving we had it in Ottawa as well. Like I thought we would just like Montreal and Toronto.
Yeah.
Even Ottawa. Yeah,
Okay.
Well, a place. So
project
maybe next year.
It may be a summer project. Okay.
Yeah. Very good. Well.
All right. Thank you, Dr. Lee. It's been a great conversation. Like I could say in your lectures that just as you're talking about all the scientific material, which is fascinating, but you also have a lot of other things to say, and it would be a fun conversation.
I'm glad you remember my lectures because I don't have notes, so it's kind of like stream of consciousness things, and so the scientific information is there, but there's much more to learn other than just the material.
Right. No, it was really great conversation, and thanks for coming to the podcast.
It's been great. Okay, it was a pleasure being here.
Thank you for listening to this conversation with Dr. Jonathan Lee. We covered topics such as cell biology, the administrative side of research, the TMM program at uOttawa, and his views on work-life balance and life in general.
This is the Dendrite Podcast. To support it, please share this episode with your friends and anyone else who might be interested. We hope you enjoyed this episode, and see you next time.